Hello there, it's Mrs. Scoville, and today we're going to be learning about non-Mendelian modes of inheritance, and we are still in genetics. So starting out, we do have 3 learning objectives. Make sure you write these into your notes. The first is to be able to define all of the modes of inheritance of genetic traits, and that's the majority of the lesson. The next is to be able to distinguish between these different modes of inheritance, and then finally, when given either a description or even a Punnett square, to identify these. You also will have to create Punnett squares with these, but we'll work on that more in class. All right, so for starters, we're just going to list all the modes of inheritance. So the first is one we've already seen, which is autosomal dominant and recessive, and that's the majority of what we've been spending time on. I do want to remind you guys that autosomal, what that is referring to is just any chromosome that's not a sex chromosome. So in humans, we have 23, so 1 through 22, those are all autosomal. The next is codominant, then incomplete dominance, sex-linked, multiple alleles, and polygenic. So 1 we already know, 2, 3, 4, 5, and 6 we'll be going through in this lesson. All right, so first of all, starting out with autosomal dominant and recessive, again, these are traits expressed in non-sex cells, and not even just non-sex cells, but on those non-sex chromosomes. And all of these are going to follow those rules of inheritance that we already outlined. So dominant and recessive, the dominant takes over, the recessive is only seen in 2 copies. It's also going to be all those traits that we can use just a Punnett square to predict those ratios. All of these examples that we talked about already, I'll show you one of these in a second. Huntington's disease also follows an autosomal dominant pattern. You can spot these because if you see a dominant gene, that's the one that's going to be expressed when you see the phenotype. So this is basically what we're used to up to this point. So a reminder of what this looks like. The Punnett square is going to be normal. It's something like this. When we saw height in plants, tall is dominant to short. So if you had a big T, well, all of it was going to be tall then. It didn't matter if you had 2 copies or 1, it was tall. So basically where we have the 2 phenotypes, tall or short, that is going to be autosomal dominant or autosomal recessive inheritance, and we're used to that. That's the normal one. Now we're going to get into some that don't quite follow these patterns, okay? Because some alleles aren't going to follow that nice pattern where, one is dominant, one's recessive. Some are neither, okay? They're neither dominant or recessive, or they're both dominant. And the other problem, too, is we've been looking at things where it's just 1 allele, 2 different versions. Many traits are controlled by more than 1 gene, and these are going to be our polygenic. So we can't just make it as simple as, big T, little t for those. So the next type that we had here that's not quite, following those normal rules is codominant. And that's pretty much what it sounds like, codominant, like they're both dominant. So for that, you have an allele, and there are different versions of it. And remember, an allele is just a version of a gene. And it turns out that there's more than 1 dominant trait for some genes. And so what this means is when it's heterozygous, when there's 2 different alleles listed, for both of the dominant alleles, that means that both of them are going to be expressed because they're equally dominant. It's like they're equally strong. So you're going to be able to spot these by looking for a 3rd phenotype. So the homozygous traits are going to be basically mixed together equally in your codominant. And if you're having trouble visualizing this, don't worry, I'll tell you in a second what to see. But just to kind of narrow down the field too, you're going to see genotypes with 2 different capital letters when you're looking for codominant as well. So an example of this actually is with roosters, and we don't see this just in roosters, but this is a pretty clear example. So in roosters, black is dominant, but white is dominant as well. So we've got a black rooster right here, white rooster right here. That trait is dominant in both of them. So if you were to cross, really it would be a hen and a rooster, but if you were to cross a BB and a WW, hen and rooster, that would result in an offspring, if we did the Punnett square, BB, WW, that have BW. And so that ends up being something called roan. It's a black and white mix. And you can see the black and white, it's, they're individually black or white, but in roan, both black and white are both expressed. You notice, though, it's not gray. You still see the black and the white. They're equally there. So that's why this is codominant. Okay, so another example of this is actually sickle cell anemia, which we've seen several times. As a reminder, it's caused by a missense mutation. But here, in sickle cell anemia, you have normal cells. In sickle cells, if you are a heterozygote, so if you have a 1 copy, 1 allele that has the sickle cell trait, but not the other one, you'll have some of each, okay? So obviously if you have the sickle cells, you're sick. And a Punnett square of what this looks like with sickle cell anemia is if you cross someone with sickle cell anemia and another who's called a carrier, that's another term for a heterozygote, so they have a normal gene and a sickle gene. break it up just like normal. Our Punnett squares are still normal. The only thing that's different is what goes into them. Again, this is codominant, so we still put capitals for both of them. In that case, you would end up with 2 out of 4 children that would have severe sickle disease, and 2 out of 4 that would be mild. Okay, the next trait here is incomplete dominance, and that's sort of what it sounds like. Oh, I'm dominant, but I'm not quite there. So here, what this will look like is the phenotype of the heterozygote, and for these, identifying them, a lot of times we're going to look for that mixed one and see how it's different. So the heterozygous genotype is going to be not only distinct from the phenotypes, but usually it's a mix of the other 2 homozygote genotypes. And so what this will look like is you're really just looking for a phenotype that is a mix. So we're going to have 3 phenotypes instead of 2 like we had with autosomal recessive and dominant, and our third phenotype is going to be in between the other 2. So it's going to be a mix. And again, in this one, we will have a genotype with 2 different capital letters, but this should be our last one. Okay, so an example of this actually comes with snapdragons, which are a flower, and there's an example of them here. You can sort of look at, you can squeeze these and they open up, but it almost, the face kind of looks like a dragon. That's why they're named that way. So R encodes for red pigment. W, that allele, codes for no pigment. So white snapdragons just don't contain any pigment. That's why they're white. A heterozygote in that case will have an R and a W. That's a heterozygote because it has a mix. It's not the same. So those basically, they only produce half the amount of red pigment, and instead they're pink. So that's what I mean by it being a mix. Red would be a homozygous. White would be homozygous as well. The heterozygote, the mix in between, is a mix of the 2, literally. Red and white make pink. So your incomplete one, it's going to have the one that's a mix. So if we do a Punnett square on these, what I was just saying there, red flower and a white flower, when you cross those, again, you just set up the Punnett square. All of those are going to be pink. This is definitely incomplete dominance because instead of being red and white separately or striped or something like that, which we would see in codominance, it's pink. Okay, so it is a mix. That's incomplete dominance. Okay, the next one, and this one's a little bit more complicated, are sex-linked traits. And all this means is that they're on sex chromosomes. So these are traits that are on the sex chromosome, so either the X or the Y. I will tell you it is usually on the X. These usually are recessive and almost always are more frequent in men because men have only 1 copy of the X. This is only going to be true for the recessives, though. And in those cases, women must have 2 copies to have the trait. So how you're going to know that this is a little bit different, when you actually write out the genotype for sex-linked traits, you have to indicate not only is it a male or female, so X or Y, but you'll also actually put the trait on the X or the Y, and so that's how it'll look different. And just a note with these, I said recessive, that's the most common one. We are going to see cases, though, where they're dominant. They actually can be codominant as well, so it gets a little more complicated. And how you can spot these, the dominant or recessive gene is going to be on the sex chromosome, and this is just cut off here. We're going to see it again in a second. Like you would write the X and then the letter as a superscript. So we'll see that in a second. Yep, right here. So an example of this, the recessive one, is with hemophilia. And hemophilia is a sex-linked recessive blood clotting disorder, and it is inherited on the X chromosome. So what I was just saying on the last slide that got cut off a little bit is you have to write the trait on the X or the Y, and in this case, it's inherited on the X, so we write an X. And then as a superscript, that's where you write the allele letter. So superscript H, that's a normal gene. This is recessive, so we still use our dominant and recessive notation. So the little h would be for hemophilia, since it's recessive. So when you're doing the Punnett square, you have to know male, female. So this is a female and a male mated together. And the mom, it looks like, is heterozygous. So she has 1 normal and 1 hemophilia. She would not be hemophiliac. She wouldn't have any blood issues because she has a normal copy. She's got 1 normal, 1 messed up. Dad is normal, but if you look at the offspring, this daughter will be normal. This son will be normal. This daughter will be what's called a carrier again. She's a heterozygote. She's got 2 different copies, but this son will be affected. So that's how we see the men are affected more with sex-linked recessive traits because they only have 1 copy of the X. All right, so moving on from dominance, from really looking at dominant ones, when sometimes we have multiple alleles. So when there's more than 2 alleles for a trait, so not just red or white, for example, there's going to be more than 2. And so here we're going to have at least 3 different letters. That's pretty much the easiest way to tell if you have a multiple allele. And the most common example of this is blood type in humans. Humans can be type A, type B, type AB, or type O. So for these, you write A, B, or O. A lot of times as well, they'll write an I with an A or a B as a superscript. That's just sort of a notation they use for blood. So if we look at a Punnett square of this with multiple alleles, if it's a cross mother who's O with a father who is AB, well, to be O, you have to have 2 copies of O. And AB, well, that's pretty clear. It would be A and B. So you can do that, A and B, O, O. You write them next to each other. So how this works, with the genotypic ratio, you'd have 2AO, 2BO. You're going to have 2 different phenotypes there, 2 that are type A and 2 that are type B. The only way that you can be type O is if you have 2 copies. So here, what is the probability that the offspring will have type O blood? Well, it's 0%. So the thing that's interesting with blood type, if you haven't kind of noticed it already, but we actually have 2 examples in blood type. Not only is it multiple alleles, you have A, B, and O. A and B are codominant to each other. So you've got multiple situations going on in this one. But multiple alleles, again, more than 2 letters. So A, B, and O are 3 letters. That would definitely be multiple alleles. Okay, and then our very last example here are polygenic traits. And these are going to be traits that are controlled by 2 or more genes. So again, just multiple genes control these. And how you'll be able to identify these, first of all, we're not going to be able to do a Punnett square on them, so I won't ask you to do one. But phenotypes are going to be, you're going to have a lot of them, and a lot of times they're kind of along a spectrum, so gradual spectrum. And examples of this include human height, so you can see people get gradually taller, shorter, and these are also going to be traits that don't have nice, easy predictions. So if your parents are tall, you might be short. Okay, if there's somebody that's short in your family, you can't just look and say, oh, you're going to be tall because your parents were tall. So these are things that are kind of a little bit more complicated because there are multiple genes involved. Another example is eye color. You can see there's a whole spectrum of eye colors. And I think you probably know people can have from very light blue to green to kind of hazel or really dark brown. All kinds of eye colors that are different. And then probably the most dramatic example over here is skin color. This really helps you see that this is a very dramatic trait that's controlled by multiple genes. This is one family. And these 2 little girls right here are twins that were born at the same time, and these are their parents. You can see these parents are somewhat light-skinned, and because these traits are controlled by so many different genes, they had 2 twins, one that has, kind of caramel-colored skin here, and one that's very white. These are the same. These are from the same parents. And because those parents have a lot of light skin in them, this daughter came out white because skin color is just a polygenic trait. So it really just depends what genes are expressed for those traits. Okay, so with that, we are at the end of the lesson. Make sure you write your complete summary and complete the practice questions as well.